Could a cure for cancer be closer than you think? Welcome to Live from Stage 4, where NBC takes center stage as we talk to experts, share inspiring stories, break down science, and shine the spotlight on what matters most. Because when it comes down to it, the spot, for us and by us, is all about us. Welcome to Live from Stage 4. In today's episode, we're tackling the urgent and deeply personal topic of federal funding cuts and their impact on cancer research. It's not news to anyone that research progress matters. One study cited in the Journal of Clinical Oncology found that every $300 spent by the government on cancer research extends someone's life by 1 year. We're living in a very exciting and productive time for cancer science, but grant cancellations have forced labs to lay off staff, stop studies, and push promising researchers to abandon projects. Because early stage, risky research is mostly publicly funded, cutting it now will slow future breakthroughs and clinical advances for years. The administration argues the system is bloated and needs reform. But critics say there is no clear plan to replace the lost funding or preserve the research pipeline. Our guest is Dr. Gloria Echeverria, a molecular biologist and passionate advocate for scientific discovery, who leads her own lab at Baylor College of Medicine. Gloria's research centers on the triple-negative breast cancer. As we know painfully too well, it's an aggressive and hard-to-treat subtype of breast cancer that lacks effective targeted therapies and often affects young women. Her lab at Baylor College investigates the molecular and genetic mechanisms that make TNBC resistant to standard treatments like chemotherapy and radiation. She shares what it's like to run a research lab in today's climate, the challenges of securing funding, and the ripple effects these cuts have on training the next generation of scientists, advancing new therapies, and supporting patient advocates. Join us as we explore the real-world consequences of policy decisions, the resilience of the scientific community, and the hope that drives researchers like Gloria to keep pushing forward, even when the future feels uncertain. Let's get started. I am so incredibly excited to have you here with us.
Thank you for the opportunity to be here. This is wonderful.
All right. Before we delve into the federal funding cuts and the real world impact of those cuts, I want to set the stage. And because our podcast is live from stage 4, set the stage is the right thing to do. So tell us about your background. We want to know what you do at Baylor College of Medicine.
Thank you so much. We'll set the stage. My name's Gloria Echeverria. I got my PhD at Baylor College of Medicine, where I now run my lab. In between, I did a postdoctoral fellowship training at MD Anderson here in Houston, Texas, where I fell in love with triple-negative breast cancer research. It ignited my passion for helping people with passion for science. And I have since then focused pretty much exclusively on TNBC in my research. So I'm a PhD, not an MD, so I do science research in a laboratory setting, right? Not in the clinic. And I started my lab here at Baylor College of Medicine in Houston in 2020. And so it's been about 5.5 years that I've had my own lab. It's also important that I mention that I'm speaking today on behalf of just myself. So everything I'm saying is just my own views and not an official representation of my institution or funding sources or anything like that. We are a basic molecular biology lab that studies translational questions pertaining to triple-negative breast cancer, TNBC. So there's not a single project in the lab that's working on something that I don't think could help TNBC patients one day. That is the only goal. Our job is to study the biology, the molecular mechanisms, the genetics that make triple negative breast cancer tick so that we can devise better ways to therapeutically help women living with TNBC to improve their outcomes. As many of the listeners here may know, TNBC, as opposed to some other breast cancer subtypes, can preferentially affect younger women. It has a deficiency in options for targeted therapies, molecularly targeted therapies, right? So patients are left with few options for treatments, and really there's not a lot of personalized therapy at all that's currently approved in TNBC. We're trying to fill that void and trying to understand in depth molecularly what's going on in TNBCs that are resistant to our standard of care therapies, chemotherapies, radiation therapies, et cetera. The way that we do this is by focusing on a compartment in the cell called the mitochondria. Many of you may have heard that you get your mitochondrial DNA from your maternal lineage. You may have heard that mitochondria are the powerhouse of the cell, which is true. Mitochondria are very interesting compartments in the cell that do generate energy that the cell needs to survive, and they generate a lot of other molecules that help the cell do what it needs to do. And we've discovered that TNBCs can alter the way their mitochondria work, especially in response to stressful chemotherapy or radiotherapy exposure. And so we can take advantage of that. We can harness that elevated dependence on the mitochondria for our own benefit. And so what we're doing is working on understanding molecularly how that mitochondrial change happens so that we can use drugs to block it and do a better job of killing cells that are resistant to standard of care therapies.
This is fascinating. And I have to just say in parentheses, my husband and I have been watching Law and Order SVU lately. The word mitochondria comes up every single episode. I think I'm going to play a drinking game at this point.
That is so funny.
It's one of those things that you hear all the time, but don't really understand. So triple negative breast cancer is, I guess there are about 15% of people affected by triple negative breast cancer. Mm-hmm. And the type is really not the type. It's the type of what is not, right? Exactly. I mean, if we talk about the other types of breast cancer, the ER-positive hormone-driven cancers, HER2-positive cancers driven by HER2, this is sort of a basket of what is not, basket of different diseases that are not driven by hormones or something that we know. Mm-hmm. And of course, what you do is of utmost importance, right? Because you are looking at where it starts. What is triple negative? What are the different things that drive triple negative cancer? Mm-hmm. So let's talk specifics. Paint us a picture of your lab. What is it that you do on a normal day? And what are the priorities right now on the stuff you're working?
Mm-hmm. Yeah, that's a really good question. So not to make everyone jealous, this is my dream job. I love my job. I get to do my favorite thing in the world every day, which is learn something new, try something new, and to work with passionate, dedicated, creative people. So who is in the lab? I am so pleased and proud of the people in my lab. We've got people at a lot of different levels. So we've got postdoctoral fellows. So these are people who have completed their PhD training and are a postdoctoral fellowship is akin to like medical residency training, but on the lab side. So it's sort of the stepping stone between PhD and a lot of jobs that come next. I also have a few PhD students. So these are people who have completed their undergrads. Sometimes they've completed master's degrees, not always. And they're working on their doctorate degrees. And I also have a couple of staff scientists, so people at different levels who are not necessarily working on a degree or a fellowship in a formal training program like that, but they are scientists doing research in the lab. I'm also very privileged to have a few bright-eyed, bushy-tailed undergraduate students from Rice University, which is across the street from the Texas Medical Center, who come and in their spare time when they're not studying for their undergrad courses, they come and do some research in the lab to get experience. So that's who we are. I would say on my average day, I am in my office. So unfortunately, I don't get to pipette and do the physical work anymore. I just can't be in the lab doing the experiments myself. But my role is to get grants to support the lab, to mentor and oversee and guide projects and the trainees in the lab, to help publish, present our work in oral and written formats, and to support the education and training of the next generation. So that's what takes up my time. I love it. It's so fun. Being in an academic institution, a huge part of your job is teaching, mentoring, whatever words you want to use for it. So teaching outside of the classroom. And I love working with trainees. It's just really rewarding to see them grow and learn to love the science that they do. And I would say the main time-consuming part of my job is trying to get money to support all of the wonderful people in the lab, to support their salaries, to support the supplies and reagents and equipment that we need to do the research.
So this particular period in time is very difficult for what you do. So how do you manage to keep your doors open amidst all of this funding cuts, uncertainty?
Yeah, it depends on when you ask me. I'd be lying if I told you I'm not worried or stressed about it. Of course I am. I don't know of anyone in this line of work that isn't concerned. But I am so fortunate to get to work on a disease I care about, to feel like I'm helping maybe in small ways eventually, hopefully, to get to train young people, to get to think about science, learn things. I learn new things every day, right? Because the nature of research, we're generating new knowledge that nobody knows, that doesn't exist on planet Earth. It's so fun. So that is what gets me through it. And then speaking with patients and advocates like yourself, it's impossible not to be motivated. So even though there are a lot of walls being put up and barriers and divisiveness that's happening, no matter what a politician says or an institution says or what funding comes and goes, the content of the work doesn't change. And the importance of the work doesn't change. If anything, it gets more important with every day that goes by. Our job is incredibly important, and it's urgent that we make headway. Maybe we're not developing a new drug every day. Things aren't getting into clinical trials every day, but research is like a brick wall. Everything builds on other things. So it's very motivating to get to work with patients who remind us that what we're doing really might help people one day.
Do you get to actually work with a lot of patients? Because this is one complaint I've heard over and over again from people who do basic and translational science, that they don't get to see the actual patients.
Yeah, so that's a good question. I'm not seeing patients at all through my formal job, right? Because I'm not a clinician. I do collaborate with a lot of clinicians. I have wonderful collaborations with breast surgeons, breast med oncs, radiation oncologists, pathologists. So I do have a connection to the clinic in that way. Now, my interactions with patient advocates are all through wonderful advocacy networks, like what you're involved in, NBC Alliance. We have wonderful patient advocacy representation through passionate people like Mr. Josh Newby here at Baylor. Yeah, at Baylor, we're fairly spoiled. We have the importance of advocates in our face, at the forefront of our attention all the time. That's certainly not the case at smaller institutions. We kind of have a privilege here of always having advocates that we have contact with and can reach out to. Not everybody does, but I think what you're bringing up is a really important and consistent issue that's been brought up in recent years, that there is a disconnect in access and communication between researchers and advocates. And so there have been some studies, poster presentations at conferences, publications addressing like how do we overcome these barriers. So I think we're moving in the right direction, but certainly we're not there yet.
I was going to ask this question later, but we were talking about your daily work, and one of the things you do, and people who work in your lab are postdocs and even undergraduates, I'm glad to hear, and that participate in your work. But I've also been reading quite a lot about how it's so difficult for those who graduate right now to find jobs. And I'm sure it's heartbreaking for you. And what advice do you give them?
The thing that I've witnessed with the new kind of climate with research that we've been experiencing in the last several months, I see people at all stages getting affected. The graduate school admissions process this past spring, so for students who would be starting this August right now, was an absolute mess. And honestly traumatic for many people involved. As you may be aware, a lot of institutions in the country had to reduce the number of new students they could take because of worries about future funding, right? Which is, it just makes it more competitive, it makes it more difficult. Many institutions had to rescind offers that had already been given out because of new financial worries that arose in the interim. And that's just something really awful to have to do to a young person at the very beginning of their scientific career, right? It's happening at every level. So PhD students who are graduating, who might be looking for postdocs at academic institutions, are having a really hard time finding positions. I want to take a new postdoc, and it's very daunting to convince myself that it's financially wise to do. As you and I discussed previously, it's also happening at the transition from postdoc to faculty. There are a lot of real examples, some of them from wonderful people here at Baylor and friends I have at institutions around the country, of faculty job offers being rescinded, put on hold indefinitely, job openings being taken away. So it's not that everything's gone, it's that things are much more difficult and much more limited. Now, your question was, what do I tell people? All of those roadblocks don't mean your science isn't important. They don't mean that your science isn't interesting and breaking new ground. Doesn't mean that your science doesn't matter for breast cancer patients. It just means you're going to have to put up with a little bit more difficulties to make it happen. And it might look different than you had pictured in your head. The path may not be as linear as we used to be able to follow. The people that I'm thinking of that have had these hardships thrown at them are really tough and really love their science. And I haven't seen a single one yet throw their hands up and leave and give up. Now, that's kind of the thing, I think, and part of what makes the attitudes towards research and the climate towards research quite disheartening and confusing and unfounded, to me at least, that scientists love what they do and they really care. And it's already a pretty thankless job, right? Scientists work long hours. We put up with rejection from scientific journals, from grants, applications all the time. And research is hard. By nature, you're developing new knowledge, right? So a lot of things won't go the way you thought they should, or methods won't work, and we have to kind of pivot and troubleshoot.
It's very competitive too.
Competitive, and it's a rocky road. And so the people that are doing it really love it and are used to putting up with difficulty. So I guess we're a little well prepared. The new difficulties that are being thrown in people's paths are another level that hasn't been experienced before, and making something that was already challenging, making it a lot more scary and unpleasant, especially for our young people. But I don't see people throwing up their hands and just leaving science, at least in my personal observations yet. We're still early on, because I think scientists, we're hard-headed and we love what we do, and we'll put up with a lot to be able to do the work that we want to do. Now, with that said, in the field, I say there's a lot of concern of brain drain, and a lot of European institutions, for example, are recruiting. And, I do worry that at some of our early levels of training, that maybe like undergraduates who are considering, maybe grad school, maybe med school, maybe no school, right? Maybe they'll think a little bit less positively about graduate school in this time. I can't imagine that won't happen to some degree, especially the longer that goes on.
I think you're right. All right, so let's get back to actually concrete examples. So I know that you've had to reevaluate the priorities of your projects in your lab, and you had to scale back and put some projects on hold. Can you give us an example or a couple of examples?
Yeah, absolutely. One example is any health disparities, not only socioeconomic, but also real genetic and biological, concrete scientific differences in cancers, including breast cancer, in patients from different backgrounds, different descents, right? And those really matter for clinical trial design. They really matter for outcomes and everything. And it's a huge area of study, and rightly so. And that work it's gone from federal funding. It's gone. I have a lot of friends who directly focus on cancer health disparities, and those grants are canceled, non-existent anymore. I have one NIH grant right now in the lab that is not disparity is related, but it's a breast cancer focused grant, a breast cancer molecular biology grant focused on the research I was talking about earlier. There used to be funding opportunities to expand your grant to address health disparities in the light of what you had proposed, right? So basically, this grant is looking at mitochondrial function in TNBC. Well, now I'm going to expand it a little more and compare TNBCs from Black women and from white women. And so I've written proposals for things like that have the mechanisms seem to not exist anymore, and the application isn't being reviewed, right? As far as other personal experiences, many of you might remember earlier in this spring of 2025, there was a huge interruption to the grant review process.
Right.
I witnessed the problems that arose from that from the standpoint of an applicant and of a reviewer. Many researchers, such as myself, are peer reviewers for the NIH or NSF. Peer review is the backbone of research. It's the best we have right now in the scientific system. There were really last minute and chaotic delays that thankfully eventually picked up. For the past couple of months, grant reviews have been proceeding, and grants are, being reviewed, and scores are being returned to researchers. And some of the actual awarding of funds has been delayed, has been in the news a lot lately. As of, I think, yesterday, the delay was blocked. And so people are starting to get their money. So there are some little glimmers of hope as well. But I do want to mention the point, this kind of illustrates the point that things are changing constantly. I would say I can't go 2 or 3 days without some new big news directly relating to my work and my field. And that's ignoring all of the other big news in our society. Just about my personal career field, every couple of days, something new happening. It's very distracting, right? And unsettling.
I hear you. And I'm glad to learn from you that things are finally improving. I have to tell you that in the past, I was a little less conscientious about doing research before interviews. I used to wing it. But I think this is so important, and this topic is so important that I actually have done some homework. I've done some research, and I read a piece that you wrote for MetaViva, and it was great. I would welcome our listeners to go to our page for this episode and look at the resource section and check out the article itself. I think it's absolutely worth reading. And in this particular piece, you mentioned that we've all heard this by now, that the funding cuts are targeting overhead or indirect costs. So for most of us, this sounds kind of unimportant in a way, and just administrative stuff. What does it matter? But I think it's worth asking you to explain exactly how this seemingly unimportant item is actually essential.
I totally understand how, from the outside, it seems obvious. Well, yeah, that's where you cut. That's how you save money. We're not here to support a building, a college. We're here to support research, right? So I get it. I get that sentiment. The funding cuts or impending potential funding cuts are kind of twofold. There's, in general, an overall reduction of the NIH budget that's being proposed in the new federal fiscal year that starts on October 1 that would dramatically decrease NIH funding. And that is a big part of what institutions are bracing for and trying to prepare for. And already we're seeing a cutting in one half the funding rate of major NIH grants right now for this cycle. So typically we call this a pay line. So typically the pay line for NCI past several years has been 9 to 10%, meaning that 90% of submitted grants do not get selected for funding, and the other 10 do. That pay line is now, for the next cycle, the NIH put out an official post about this end of last week. This is what I mean. Everything's changing all the time. That it's going to be 4%. As a researcher, I'm currently writing an NIH grant that I'm going to submit in a couple days, and I'm questioning, why am I doing this? well, I'm doing it because I'm definitely not going to get it if I don't submit. It's an important project, but it really is extremely disheartening. And I want to emphasize this isn't specific to me. This isn't specific to my institution or breast cancer. Anybody in the biomedical research space is feeling this. So that's one angle. Just the overall funding cuts mean that fewer grants can be funded. When fewer grants are funded, it just means there's less research done, right? That's what that means. So fewer labs will exist, fewer PhD students will be trained, fewer postdocs will be trained. So there will be fewer PhDs produced in the United States. These things will take years to manifest, right? But they will happen if this is what happens. So just less research output overall is what's going to happen, which tends to really matter if you are someone who might benefit from the research, like a patient who has cancer, or someone who has cancer, right? So this affects everybody. To answer your question about indirects, they're actually negotiated rates that each institution negotiates with each funding agency. Each institution negotiates a specific rate with NIH, NSF, State of Texas funding, and the rate is different at every institution. A lot of places, especially medical schools that don't bring in big tuition costs from thousands of undergraduate, like University of Texas, where there's however many tens of thousands of students in medical schools, which I'm at one, we don't have those student bodies, and indirects from research funding are a lot of what help support the institution. So why does anybody want to support the institution? Well, it's what enables us to do the research. So it sounds silly, but the money goes to keep the lights on, to buy common specialized equipment that's too expensive for a lab to buy. Some of the routine equipment that we use costs $100,000, $500,000. So no individual lab has those things. Rather, institutional cores, as we like to call them, for example, will have really specialized equipment like that people like me can use as like a fee for service. So equipment funding, service contracts for the equipment, funding scientists to specialize in and run the equipment. So things like that. Administration. We have to have HR. We have to have payroll. These are the things that make the institution run. And without these things, there would have to be a huge scaling down. Right now, a lot of institutions, their indirect cost rate is up to 60% of the grant amount. So let's say I got awarded a $1 million grant from the NIH, $600,000 on top of that would be given to my institution, which I agree is a ton of money. I think there are a lot of reasons for that rate. It has to do with the locale. If you're in a bigger city, the rent for the building is more expensive than if you're in a rural place. Everything like that has to do with indirects. The sentiment I hear from a lot of people is the systems can always be optimized, right? No one would say, oh, our academic research system is flawless. We shouldn't change a thing, right? But the incredible abruptness, the quickness of these changes is unmanageable for places. These are huge changes that I can imagine might be figure outable over the course of a few years. Like, here's the plan. All these places need to figure out how to make changes to reduce indirects by X% in 5 years. That's not what's happening. It's, we're going to flatly decrease it to 15% everywhere. Hopefully it won't be that drastic. We still don't know, right? For a place like a medical school where the indirect rate is 60%, going down to 15% would be pretty dramatic. This is why places are scared. Institutions are scared, and then individuals like me are scared too. So the message is the indirects are really important. We cannot do our research without them, unfortunately.
Yeah. It's clear, even for somebody like me, a little person who is doing a podcast, you would think, what's the big deal? You turn on Zoom and that's it. But in fact, there's a lot that goes into making a podcast, and those are indirect costs.
I'll interrupt this wonderful interview to give you a quick and funny look. A tiny science detour from the British Society for Cell Biology. Mitochondria are organelles that are virtually cells within a cell. They probably originated billions of years ago when a bacterial cell was engulfed when visiting what was to become a host cell. The bacterial cell was not digested and stayed on in symbiotic relationship. A true story, isn't it? Of a visitor that stayed on and on and on forever. The mitochondrion has certainly made sure its presence is felt. The mitochondria reproduce within the host cell. These visitors have become so essential to the life. They provide most of the chemical energy and death that medical specialists are actively introducing them into egg cells. This is being done as part of a protocol in the treatment of infertility in humans. It is interesting to reflect, isn't it, on the fact that the organelle that once entered the cell by accident is now being captured and transferred by humans and placed in the egg cells of another human being. And now back to our conversation with Dr. Echeverria. Let's hope with a little extra cellular energy.
Well, so I hear that there is a big shakeup in federal funding. And again, from my homework, I read that your lab is supported by the range of organizations, right? And NIH is one of them, obviously, but then American Cancer Society, Mary Kay Ash Foundation. So maybe you could give us a little overview how, and I think we've already pretty much covered it, but why, in your opinion, diversifying your funding sources is so essential for your work?
Yeah, thank you. I guess in the past, I never would have thought, oh, I better diversify on purpose just in case something completely falls through with one of these funding sources. That would have never entered my mind. These are critical institutions that have been around in our country and they're not going to change. And now we're seeing that the things that we used to be able to count on, maybe we can't count on them anymore. I mean, we're being told to be strategic by our supervisors, by our institutions, like you've got to start diversifying. We've got to reduce our reliance on the NIH or the NSF, right? So that's what we have to do now. Now, I do have a fairly diversified funding portfolio right now. It wasn't necessarily on purpose. It is that way because I've been pretty proactive. I still consider myself a fairly new lab, so I'm sort of in a growth phase and trying to be able to support more trainees and more projects. So that has required me to be very vigorously applying for a lot of grants. So I wouldn't say the diversifying was like on purpose and strategic, but I think now it absolutely is for people. It's going to be completely necessary. So now more than ever, foundations like Mediviver, American Cancer Society, Komen, Mary Kay Ash, AACR, that depend on donations from generous people to fund research, private foundations, they're more important than ever. And I cannot tell you how grateful I am. And researchers just like me, we are so grateful that there are people out there who want to support the work and that there are avenues for us to get some research support aside from just federal funding.
Yeah, I'm so glad that we've actually touched upon this because I think it's important for our listeners to understand that, maybe we can talk about it for a few minutes. I interviewed a while back for my old podcast, I interviewed the executive director and the founder of Twisted Pink. And their main mission is to raise funds and give grants. And I couldn't understand. We all know that to get a drug to patients takes not a few $1 million or $1 million. It takes many times more than that. And before I spoke with her, I didn't understand what is the importance of seed grants. And I think it would be interesting for our listeners to actually hear from you what it means to get a grant from Mediviver or Twisted Pink.
Yeah. There are a lot of foundations that'll give out $25,000 grant, $50,000, $10,000 grants. Research is expensive. The people that do research, it's their job. They have to have a home and food. We have to pay people salaries, right? So think about how much a salary might be. Do the math, and it adds up very quickly. I'm not trying to argue, oh, small amounts don't matter. They absolutely do. We can't do our research without contributions from these kinds of grants, right? Especially with smaller foundations, the research tends to be a little more translational, more directed towards near-term patient relevance. And so that work is really important. And a lot of times people will get a $50,000 grant for a really special project where they're going to do something that's really going to have potential to impact patients quickly. It's a 1 year of funding, and they can use that 50,000 to do a really special new method, for example, to learn something new that may not fit in the scope of a bigger federal grant. So what you donate, it's going to go to support a graduate student who is studying the proteins and the molecules that are going to be targeted by the drugs that we're trying to develop for breast cancer. The money is supporting equipment that lets us analyze tumors that are donated by patients from the clinic, and we can analyze them in new ways that have never been possible before, that might lead us to new biomarkers or new treatment strategies, right? So all of those different types of information have to be generated and put together to get a drug into a first-in-human clinical trial. I mean, there's a huge number of steps, a huge number of years that underlie this, right? And it takes just a lot of resources, unfortunately. The FDA, it's one of the shining stars of our country. Other countries model our clinical testing, our FDA model that we have here in the United States. So we're leaders in the world of how to do this, but with doing this carefully and safely and ethically, there's a lot of checks and balances and a lot of safety studies that we do. I mean, we're trying to help people not be flippant with things that we're trying to test in patients. So
absolutely,
being conscientious about all those things, I think just the nature of it requires a lot of resources to do things the right way.
Why don't we end on something more positive?
Yes, we must.
So what is the positive side that you see in cancer research in the future for us?
Yeah. The science, the breakthroughs, they're not different. It might get slower and harder, but it's not going to go away. I know promising new labs out there. My own lab has some exciting discoveries that are getting into the clinic in the next 1 to 2 years.
Well, give us an example. We're dying to hear.
Well, so some of the mitochondrial biology that I work on in the lab, we've had wonderful collaborations with small pharma entities that have enabled us to test promising new drugs, novel drugs that are not in any breast trials yet. Some of them aren't even in humans yet. And we're working with our wonderful colleagues here. We have phase 1 trial specialists, breast medical oncologists, drug development specialists, and working with wonderfully talented people at the pharma companies. And we're getting these things through teamwork and collaboration. We're getting these things towards a clinical trial, which to me is the ultimate success. So once I do that in a few years, then I'll retire.
That's right. That's right.
I'm joking.
Once you find a cure for triple negative breast cancer, you can retire.
Put myself out of a job, right? But I see the amazing new insights that basic research is generating, and there's a lot of exciting ways that those findings are being translated. So I absolutely have hope in the continued progress of treatments for breast cancer. As many of you know who may attend conferences like ASCO, San Antonio, et cetera, there's a lot of new stuff coming out all the time, an overwhelming amount of new drugs, new trials, right? So we're constantly evolving and coming up with better treatments for patients. I don't think that will stop. Hopefully it won't slow down substantially, but I think, again, what I tell people in the lab is the work doesn't become unimportant. This work has to get done as soon as we can make it happen. And there's no circumstance or change that's going to happen in the outside world that's going to make this work not need to be done. There's nothing that's going to change the fact that women with breast cancer need better treatments. So we need to keep going, and we absolutely will.
Thank you so very much for being here. It was such a pleasure to meet you. I hope we will see you in the future. I'm sure we'll come up with some topics that maybe are more science specific that you could come and talk to us about.
I would love that.
This was Dr. Gloria Echeverria, who I can assure you will be counted as one of my favorite guests. And before we go, I want to make sure that what lab is working on has come across clearly in this episode, because I have absolutely no doubt that their research will prove to be groundbreaking in the treatment of triple-negative breast cancer. A key focus of the team's work is understanding how TNBC cells alter their mitochondria to survive under stress from therapy, which includes radiation, chemotherapy, and other types of therapy. Just to remind you, mitochondria are essential cellular power plants responsible for converting food into energy for the cell. By studying these mitochondrial changes, Dr. Echeverria's team aims to identify new vulnerabilities in cancer cells and develop drugs that can block these survival pathways, ultimately improving outcomes for patients with TNBC. Her research is highly translational, with the goal of moving discoveries from the lab into clinical trials and eventually better treatments for patients. Thank you. Thank you for joining us for this insightful conversation with Dr. Gloria Echeverria. We discussed the critical importance of research funding, the resilience of scientists in the face of uncertainty, and the promising breakthroughs on the horizon for breast cancer treatment. Despite the challenges, Gloria's optimism and dedication shine through, reminding us that every contribution, big or small, hopefully big, matters in the fight against cancer. Stay tuned for more episodes, and don't forget to check out the resources mentioned in today's episode. They will be in our episode notes on our website at livefromstage4.org. Thanks for listening. If you enjoyed the episode, subscribe and leave a review. It really helps. Follow us on social media at Live from Stage 4 and visit our website at www.livefromstage4.org for show notes and links. This episode was hosted and produced by Victoria Goldberg. Huge thanks to Ellen Landsberger, Martha Carlson, Judy Perkins, Linda Weatherby, Jill Tirabassi, and Alexis Desai for helping get this podcast off the ground. Special thanks to James Burrows, the talented composer who created our musical intro. James, your music sets the perfect tone for every episode. We appreciate you. Take care until we meet next again in 2 weeks.